Okay, so while we wait for more to join, I'm going to go ahead and give a quick introduction. I'd like to welcome everyone to today's webinar. I'm actually very excited to kind of learn more and discuss progress with our antibody therapeutic research teams to provide insights into our internal R&D program, which is designed to uncover novel antibodies for future therapeutic development. So I'm Jenna Frame from Biocytogen Boston's marketing team. I'll be moderating today's webinar and Q&A discussion. Next, I'd like to introduce today's speaker, Dr. Frank Ahn. And Dr. Ahn is the Senior Director of Antibody Therapeutics, where his key responsibilities include overseeing Biocytogen's internal antibody discovery programs, including portfolio strategies, target selection, and project implementation and management. Dr. Ahn has 2 decades of experience in biotech and has a strong expertise in pharmacology, pharmacodynamics, and cell-based and biochemical assays used to advance multiple drug modalities, including small molecules, peptides, and antibodies. So if this is your first time joining our webinars, if you'd like to ask a question, the Q&A box is open, and Dr. Ahn will address questions after the presentation concludes. So without further delay, I'd like to invite Dr. Ahn to begin his presentation.
Thanks, Jenna, for the kind introduction, and good afternoon, everyone, and welcome, and thanks for coming to our seminar. Today, I'm going to talk about a quite unique class of antibodies called the TCR-mimic antibodies, or short for TCRm, and their applications in both antibody protein or the T-cell based cell therapy applications. So here's the agenda for today's presentation. First, I would like to give a brief introduction to Biocytogen's antibody discovery platforms, and then I'm going to hit the topic of why we're interested in TCRMs and how we conduct TCRm research at the Biocytogen. Then I'm going to focus the rest of my talk on 3 case studies that highlights some of the unique features of our TCR-mimic antibody research. So first, Biocytogen and its antibody discovery platforms. So Biocytogen is a public biotech company with over 1,300 employees, headquartered in Beijing, with offices and animal facilities in Jiangsu province, office in Shanghai, and San Francisco. We have a San Francisco, US. We have a BD office, and also we have a significant CRO facility and BD team in Boston. We also have a BD group in Heidelberg, Germany. So we are a clinical stage company. We have 5 antibody assets in clinical trials. Most of them are IO, immuno-oncology assets. We are doing a lot of early stage antibody discovery with an initiative codenamed Project Integrum that you will hear in just a few slides. And we are very active in collaborations and forming partnerships. And so far, we have formed 50 molecule licensing agreement and 42 technology platform licensing agreements. At Biocytogen, we study a number of modalities for drug discovery. We have a monoclonal antibody modality, bispecific antibodies, bispecific ADCs, and nanobody. These are the single chain or heavy chain only or single domain antibodies. And very exciting that recently we also generated the fully human TCR mice in our company. So these mice are very useful for generating novel T-cell receptors for TCR-based therapies. Well, also, as I mentioned, we have a TCR-mimic antibody platform, which is the main topic of today, and we also have a GPCR platform. So all of our antibody discoveries are predicated on these 3 different mouse strains collectively known as Ren mice. So the first version is called the RenMab, that is have the fully human both heavy chain and the light chain sequences. And so these mice and this format is very good for monoclonal antibody discovery, fully human monoclonal antibody discovery. Then the next version is called the RenLight. This is a common light chain platform. Here you have 2 different heavy chains here, but 2 different heavy chains here, but you have the common light chain, like the shaded gray colors. So when you co-express these 2 different heavy chains and the common light chain, it's very easy to form the correct bispecific antibodies inside cells. So this format, the light format, is great for forming bispecific antibodies or multispecific antibodies. And the Ren Nano is the 3rd and the most recent addition of Ren mice. Oh, by the way, Ren in Chinese means human, so that's why we named this series Ren mice. That stands for fully human antibodies in those transgenic mice background. So Ren Nano was the most recent addition, and what it is that it is a single domain antibody that contains only the heavy chain V region. Okay, it's a small, it's a versatile, and can be used as building blocks for multispecific antibodies or function as its own. So there's several advantages of our these fully human transgenic mice versus other products in this category on the market. And I just want to highlight one feature is that we introduced knockouts, okay, in these Ren mice, either in all 3 different versions of Ren mice. This is to break the immune tolerance when you have to immunize the mice with a protein that is highly homologous or preserved between mouse and humans, so that more differentiating epitopes, okay, could be obtained by immunizing these knockout mice that will not be available in the wild-type mice. I would like to say a few words about the project Integrum. As I mentioned just now, this is an early stage antibody discovery initiative that is a very ambitious program where we wanted to discover antibodies targeting over 1,000 targets. So we were motivated by the gap between the large number of druggable extracellular targets and the targets actually used or targeted by the approved drugs. So there, we wanted to discover antibodies against more of the druggable targets to fill this clinical pipeline. Project Integrum has a very broad scope. It covers a very diverse target. You can from the mechanism of action perspective, or from the different diseases perspective. It has been a very successful and rewarding program so far for us. For example, we have identified hits, antibody hits against over 900 targets, and we have as I mentioned, we have 50 antibody licensing partnerships with biotech farmers or smaller biotech companies. Okay, with that introduction, I'm going to turn to today's main topic, the TCR-mimic antibodies. So the first question is, why are we interested in TCR-mimic antibodies? And actually, what is a TCR-mimic antibody anyway? So simply put, we're interested in TCR-mimic antibodies because we wanted to target intracellular proteins. It turns out that the most regular antibody targets the membrane-associated proteins or secreted proteins, and they are just a small percent of the entire proteome. Only about a quarter of the proteins are expressed as cell surface proteins or secreted proteins. So that's only a tip of this big iceberg, with the majority of the proteins are intracellular. So how to target these intracellular proteins? So let's first take a look at how intracellular proteins are recognized by our natural immune systems. So here you have a cytosolic protein, and in the cytosol, it will be broken down by proteases and into smaller peptides. And those smaller peptides will associate with class 1 major histology MHC molecules. And then as a complex, they went to surface and presented to our T cells. So T cells will recognize these antigens, these peptide and MHC1 major histocompatibility protein, one complex. And if there's a mutation in the peptide, or the peptide was totally foreign, that will be recognized by T cells and trigger the cellular immune response. And in T cells, the most critical molecule that is responsible for recognizing the peptide and the MHC complex is T cell receptor. So the T cell receptor, short for TCR. So the T cell receptor mimic antibody are antibodies that recognize specifically the peptide and MHC complex. MHC in human is also called pHLA, human leukocyte antigens. So TCR specially recognize this complex, okay, but not the individual components, either the peptide or the HLA themselves. And so functionally, the TCR-mimic antibodies is just like the T cell receptor. So its role is to recognize the presented antigens in a complex of HLA. So there are different forms of TCR-mimic antibodies. For example, people have done a TCRm bispecific antibodies that would help trigger T cell response when it recognizes the foreign antigens or mutated antigens from tumor cells. It could also be used as a TCRm CAR-T therapy, or just a T cell TCRm-based T cell therapy, which I'm going to show you one example towards the end of my talk. This slide shows some of the active TCR-based and the TCRm-based molecules in clinical trials. On this slide, all these examples are protein-based. Okay, so here we have two groups. On the top, there's engineered TCRs. These are all T cell off-the-shelf recombinant proteins. So engineered and affinity enhanced the TCR product, TCR engagers. Okay, and the bottom group is a group of TCR-mimic engagers. Okay, as you can see, these are some of the very popularly targeted intracellular proteins for different cancers. And they have different stages, with the most advanced one, tebentafusp, that was approved at the beginning of 2022, that targets metastatic uveal melanoma. And this is a type of cancer, melanoma in the eye, and one is metastasized into different parts of the body, mostly into liver, and the patient had a very short life expectancy. I think the life after diagnosis was usually the median life expectancy for these patients was usually less than a year. So the approval of this this molecule filled a big unmet medical need for these patients. So this is the protein-based TCR and the TCRm molecules. And here on this slide, it shows some clinically active molecules, either by TCR enhanced or engineered TCR in the as a cell therapy. Okay, so the T here stands for T cell therapy here. So you can see a similar group of intracellular targets here on the top, or targeted by these enhanced TCR T cell therapies, with the most advanced in phase 2. At the bottom is a different group of T cell therapies. They are a hybrid of antibody and a T cell receptor. And the big difference between the bottom group and the top group is that the top group targets a peptide and a specific pHLA complex, and these are targeting intracellular proteins. But at the bottom, they target actually extracellular proteins. Okay, and they are HLA independent. So they targeted these extracellular proteins in HLA independent way. You are going to show one example that our TCR-mimic, one of our TCR-mimic modalities is actually has actually combined both features in our molecule. You're going to see that towards the end of my talk. So how do we conduct TCRm research at Biocytogen? We do that with some unique features that are different from other companies or research institutions. First is that our antibodies are fully human, because we generate the TCR-mimic antibodies from this fully human antibody transgenic mice, for example, RenMab. And the second is that we introduce human HLA knock-in into these transgenic mice background. This is to reduce the recognition of the HLA molecule. If you remember, we wanted to target the peptide and HLA complex, okay, not the HLA itself. So by introducing the human HLA into the, for example, RenMab, so the RenMab mice will not generate antibodies targeting the HLA itself. And we actually have a number of human knock-ins of different haplotypes of human HLAs. And that covers over 90% of the most popular HLA haplotypes. And the second unique feature is that we have a proprietary immunization protocol that would allow us to very effectively induce immunity against the HLA and peptide complex. And the third feature, or the fourth feature actually, is that we have a very high throughput single B cell identification approach. And the workhorse for us is this beacon device that can crank out hundreds to thousands of clones that screen hundreds to thousands of clones a day for us. And then after we have the clones, we also have a rapid way to screen out those off-target clones that binds non-specifically or bind other peptide HLA complexes. So what we retain would be the specific binders for the peptide of interest complex with the right HLAs. And I have to point out that the entire process is pretty efficient, and we can generate some binders in just about 3 months, and in about 5 months we will have done the initial characterizations like binding specificities of those clones. This is to illustrate that also our TCR-mimic antibodies are quite diverse. Okay, here's how to read these 3 examples where we generated the TCR-mimic antibodies. So in the middle ring, that's the germline and the heavy chain germline usage, germline gene usage, and the outer ring is the light chain germline gene usage. So you can see the combination of these two give you a diverse picture of the germline gene usages. And we at the Biocytogen, our TCRm programs covers three different classes of molecules for oncology. So these are the tumor-associated antigens, cancer testis antigen, and proteins that carries the driver mutations of the oncogenic proteins. Okay, now let's look at some of the specific cases that would highlight some unique features I just talked about our TCRm discovery activities. The first case is WT1, which stands for Wilms tumor protein. So it is a transcriptional factor that is normally is responsible for some important processes for urogenital development, such as tissues of kidney, ovary, and testes. In adults, in normal people, its expression is restricted to just a few cell types. However, it's highly overexpressed in a number of cancers, including leukemia, for example, AML, and solid tumors. So this is a protein that can generate a short peptide. We call that RMF peptide, and it is presented by the HLA-A02 allele with a quite high affinity, 7 nanomolars for the association of the HLA and the peptide. And this molecule, WT1, was actually ranked #1 tumor antigen for translational research by NCI in 2009, based on a number of factors, including the impact of cancer growth and also considering the patient populations. So this is the WT1 TCRm or TCR-T related clinical trials landscape. So the most advanced molecule is from Roche, which is in phase 1. It's a TCR-mimic antibody CD3 bispecific engager. And the next molecule is called the ESK1. This is one of the classic molecules, actually among the earliest TCRm antibodies that was generated by Eureka, that is in preclinical stage. And there's a T-cell therapy that targets WT1 by Intellia. Okay, so we generated WT1 TCRm antibodies, and we actually, for our screen, we generated almost 90 different sequences, and we screened those sequences, and here shows a representative 10 clones in terms of their binding specificity. So first at the bottom is the T2 cells pulsed with WT1 RMF peptide. So this is the specific targets we wanted to target. And you can see there's a rightward shift, okay, of the cell populations, okay? So that indicates binding, and ESK1 was the positive control here. So this is in contrast to the naked T2 cells without any peptide pulsed here, and you can see the cell populations remain on the left, all versus the T2 cells pulsed with irrelevant peptide LMP2 here. So there's no binding for this peptide and MHC complex as well. So that shows the specificity. And if we look at the affinity, here we use the tetramer the pHLA tetramer to measure the affinity. So here the red dot is the positive control ESK1, and as you can see, our clones demonstrates very robust affinity in this assay, and almost all of our clones showed better affinity versus this benchmark molecule. So we have obtained high affinity and high specificity clones targeting WT1 peptide and pHLA-O2. So here just some granularities about the specificity with the two benchmark proteins, ESK1 and also this RG6007. So I wanted to point to your attention to two similar but different peptide from two different proteins. And as you can see here, that here is the peptide, the WT1 peptide we want to target here. So you can clearly see that ESK1 has some non-specific binding to other peptide and HLA-A2 complex, as shown by the bindings, those bars under different concentrations here. Even for RG6007, here it shows here's the wild type binding, and here is the MED13L binding. So you can see at high concentrations of the end of the RG6007, you still get some significant non-specific binding here. So this is in contrast to our peptide. We show a few representative peptides, several representative peptides here. And when we conducted this specificity test to those similar peptide along with the WT1 peptide, we did also see that a non-specific binding by ESK1 here, as indicated by this gray bar here. The orange bars are the target WT1 binding here. So we can see there's the light orange is another similar peptide here. So ESK1 was not very specific. And however, these clones like 5, 9, 14, and 13 clones showed much more clean binding profile profile specific for the WT1 peptide. Then we constructed a CD3 engager to test the cytotoxicity directed by these WT1 recognizing clones. So here we tested two cell lines. OVCAR3 is HLA-A02 plus and WT1 plus cell line. This and HCT116 is the WT1 negative cell line. So when we tested this bispecific engager using our clone WT1006, here you can see that we see robust killing in the presence of both the HLA-A02 and the WT1 indicated by this red line here. This is in contrast to the WT1 negative cell line, and there was very little killing here. So that's our clone. We also performed RG6007 cytotoxicity engager by this engager, and you can see here we did observe robust killing on the A02 and WT1 positive cell line. However, we also observed significant killing for this WT1 negative cell line. So that indicates there was, at least under these experimental conditions, there was some unspecific targeting by this molecule RG6007. Here's an in vivo study where we tested two WT1 TCR-mimic antibodies in the form of CD3 engagers, and 003 and 006, and two doses. The difference between those two clones is affinity, with 006 had stronger affinity than 003. And as you can see in this in vivo study, where we implanted the mice with a cell line that overexpressed WT1 here. And you can see relative to the controls, both engagers from the WT1 TCR-mimic antibodies showed inhibition, with the higher affinity ones showed a better inhibition. So, so we were pretty confident that our WT1 TCR-mimic engager was quite specific and also active in vivo. So then we let's turn to the second case, that is the KRAS. So KRAS has become a very hot target. This is not just because we just learned the KRAS was important driver mutations for a number of cancers. We learned that decades ago, but this is really thanks to two recent small molecules from both Amgen and Mirati, and also now BMS, that they come up with two small molecules that targets KRAS G12C mutation. So that actually turned this used to be undruggable targets druggable. So that generated a lot of excitement in the drug discovery community. So people are thinking of different ways to target KRAS. So our approach is to use, because KRAS is an intracellular protein, so our approach is to use the TCR-mimic antibody modality. So this is just okay. So KRAS is GTPase. So mutations will keep it in active form for longer. So that, you know, because KRAS is responsible for a lot of tyrosine kinase receptors and GPCRs signaling. So the active prolonged activation of KRAS would lead to prolonged signal transduction and tumorigenesis. So most of the KRAS mutations are at the position 12, which is a G glycine. There's different mutations. We're interested in this specific mutation, G12-V mutation. And this mutation occurs highest in pancreatic cancer, with about 30% patients carries this G12V mutation, and also in CRCs with high teens of patients carry this mutation. And then the next one is non-small cell lung cancer, with about low to high teens of patients populations carries this mutation. So these are all high impact cancer types. So we wanted to study how we can control the tumor growth that carries G12V mutations, G12V mutation. So this is the G, that's the sequence of KRAS. Here's a G at the 12th position. That's the wild type sequence here. And the mutations, G12V, when this G changed to V, actually was mostly presented as two short peptides, a timer and a dimer, and they are presented by AO3 or A11 HLA haplotypes. There was another haplotypes that was predicted to present KRAS, which is AO2, but people have never detected such complex, so that complex might not be real. So another feature of AO3 versus A11 presentation of either the timer of the dimer versus the timer is that the timer was presented in a much more abundant way versus the dimer. So most of our research is focused on the timer peptide. Here's the landscape of TCR-based or TCRm-based clinical trials. The most advanced is in phase 1-2 that targets CRC and pancreatic cancer. And a quick look is that most of them so far are on the A11 presentation of this mutation. This is in part because it was just practically easier to generate either TCR molecules or TCR-mimic antibodies that are against the A11 versus AO3. But there's a clone that TCR-mimic antibody generated by the Johns Hopkins University researchers, and that is actually our benchmark of all positive controls antibody. So Biocytogen's G12V, we have two programs, G12V AO3 and the G12V A11. So they are at the similar stages. They are being tested for in vivo efficacy studies of our clones. And here is the slide that shows the data for specific binding here. So the bottom is the timer G12V peptide. Here you can see binding. Here we use a transient transfection, so you will sometimes see two populations of cells. But when you see the right shift cells that indicate a positive signal, so you can see here is 10 representative clones for the peptide that we wanted to target. They show the positive, and here's the V2 is the Johns Hopkins peptide that is our positive control here. So this positive signal is in contrast to the negative signals from the dimer G12V peptide, or from the wild type timer peptide, or the wild type dimer peptide, or the naked AO3 HLA molecule. Here, this shows that our collection of the G12V AO3 clones have a diverse usage of both the heavy chain immunoglobulin variable region or the light chain variable region in terms of germline gene usage. And also, if you look at another metric for diversity, which is the length of the heavy chain CDR3, you can also see a nice distribution of different lengths of heavy chain CDR3s. Look at the affinity here. That the red dot is the benchmark antibody. Again, here our antibodies showed robust affinity and higher affinity than the benchmark antibody. Here we tested further the binding specificity. As I mentioned, the binding specificity is key for TCR-mimic antibodies. So here we tested the G12V mutation against the wild type, or G12C mutation, or G12D mutation, or the naked pHLA molecule. And you can see here only we saw a positive signal with our clone only with the G12V timer peptide, but not with the G12C timer or G12D timer here. So this is similar to the behavior of the positive control antibody. However, there's a significant difference between these two antibodies in that the positive control antibody, which is a TCR-mimic antibody, that also cross-reacts with the A11 presented timers of G12V peptide. And none of our antibodies recognizes this A11 presented G12V peptide. So this is just shows that in a sense that our antibodies, our A3 antibodies, A3 G12V antibodies, TCR-mimic antibodies are more specific, okay, than the positive controls. Here again, we're trying to work on the specificity. Here is that first is that we wanted to show that here's the benchmark V2. Well, we did alanine scan mutagenesis here, and you can see just a different, for example, muted this V3 to alanine, it removed, abolished the binding here. But overall, you can see that our clones, two clones, show the very different binding patterns versus the V2, and they also different among themselves. Then, based on the important amino acids, the location of amino acids, we identified a series of similar peptides from another group of GTPases. So then when we tested the binding specificity of these similar peptides presented by L3, first let's look at the positive control. You can see that here is the targeted peptide HLA complex, but however, you also got a specific binding from a different peptides from the RAB7B molecule here. That's for the positive control. If you look at these three clones, they actually showed much cleaner binding profiles with the binding only to the targeted G12V peptide presented by HLA-A03. This is then we constructed the TCR-mimic antibody CD3 engager to test the activity of these G12V L3 antibodies. And you can see here that our antibody showed robust cell lysis or cytotoxicity in the form of CD3 T cell engagers, stronger in this particular configuration than the positive controls, which is the blue line here. And we also showed that the activity we observed was specific to G12V positive cell lines. And when we tested these two constructs, antibodies in the G12V negative cell line, we couldn't observe any significant signal. So that it illustrates the killing was specific to G12V. Here is okay. Now that was all about G12V AO3. Here, just using this one slide, I wanted to show you that we also obtained a number of G12V A11, a number of TCR-mimic antibodies targeting G12V presented by A11. Here just shows that the binding specificity of these clones were very good, and also these clones don't cross-react with the AO3 presented G12V molecules. So the last case study of our TCR-mimic program is targeting this intracellular antigen, NY-ESO1. This molecule, NY-ESO1, is actually highly expressed in a number of solid and soft tissue tumors and solid tumors. So most prominently, they are expressed in some form of liposarcomas here, and also neuroblastoma, with over 80% of patients overexpress this molecule, which is this is actually a cancer testis antigen. So this molecule is presented by 3 different HLA alleles, and the majority of that is AL2. AL2, by the way, is the most frequent HLA alleles in humans. So this is the peptide presented by AL2 for the NY-ESO1. And the clinical landscape targeting this molecule is also quite active, with the GSK's and T-cell therapy is in phase 2. And another program from China is also in phase 2 here. And you notice that all here, these are TCR-T cell therapies here. And so this is also the route that we're taking here. So we immunized the HLA AO2 knock-in map mice, and we used Beacon to score the positive clones. We did clone characterizations, and I would like to show you our design of how to use these TCR-mimic antibodies for T-cell based therapy. So in contrast to WT1 or the KRAS, the modality there were TCR-mimic CD3 engagers. So this was protein-based for those two, the previous cases. Now we are looking at, using the TCR-mimic antibodies to do a T-cell based cell therapy. So how do we do that? So essentially, we actually made a hybrid of the TCR-mimic antibody and the T-cell receptor itself. So this is the normal T-cell receptor with the alpha and beta chains here. So what we did was to replace the V-alpha, the variable region of the alpha chain, and the variable region of the beta chain with heavy chain and the light chain of the TCR-mimic antibody. So the resulting molecule is a hybrid of an antibody part that is responsible for recognition of the antigen and HLA complex. So this is different from the normal TCR T cell that uses the V alpha and the V beta as the recognition unit, or the CAR Ts, which we are quite familiar, that uses a single antibody motif to recognize the antigen. Structurally, however, our TCRm-T, okay, this structure is similar to another structure that was discovered by a group of researchers in Tsinghua University. So they call that a STAR-T. So structurally, they are very similar. However, functionally, they're different. Here, okay, the STAR-T recognizes extracellular targets. So the recognition is HLA independent. And our TCRm T cell therapy recognizes the intracellular target, and the recognition is HLA restricted or dependent. So here that we tested the binding specificity of the NY-ESO-1 peptide, and you can see a nice rightward shift that indicates the positive signal for these 3 representative clones here. And this is in contrast to the naked HLA-A2 only cells without any peptide, or the HLA-2 complex with a relevant peptide here. Here again, you can see the affinity was robust for these clones. With our affinity is similar to the positive control, which is also from Johns Hopkins University, clone V10. Then to study the specificity in more granularity, we again did the aniline scan mutagenesis. And here, the different color, the heat map, the lighter color indicates abolishment of binding when you mutate that position into alanine. So you can see that the middle W is very important, and but also the clones shows a different patterns in terms of different important locations. But it seems that in the middle location, these 3 amino acids are most critical for binding of our TCR-mimic antibodies to this NY-ESO-1 peptide and HLA-O2 complex. So based on that, we identified a series of similar peptides from the protein database, and we tested the specificity here. And as you can see that the positive controls showed significant binding in addition to, this is the target peptide, to other similar peptides here. So this is in sharp contrast to our clones that showed robust binding of the targeted peptide, but quite clean profile to the other non-specific peptide, similar but different peptide here. Then here is that, and then we tested the function and also specificity of our ESO1 AO2 antibodies. In order to do that, because it's a cell-based therapy, we first transduced our TCRm construct into Jurkat cells. So here shows the binding. The reporter activity. Okay, the reporter activity. One is okay. So the reporter activity is that once the Jurkat cells is activated by the targeted cells, it will generate a luciferase signal. So if you see a luciferase signal here, then that indicates the TCR-m modified T-cell receptor was working, became activated. So here we use the peptide instead of the wild type peptide, which is NY-ESO peptide that was not stable. We used a modified peptide called NY-E9V. This is also a well-accepted peptide in the community to study NY-ESO. So as you can see that versus the mock transduced Jurkat cells, our TCR-m transduced Jurkat cells shows robust killing only for the targeted peptide, but not for the similar, other similar but different peptide. And we also demonstrate here is the TCR-mimic antibody transduced primary CD8 T cells. And here we show the killing in the presence of AO2 and also the peptide. Here, two different cell lines. You can see this is a ET ratio dependent killing. With the higher ET ratio, the more killing of target cells. And this is in contrast to if you have NY-ESO negative cell line, then you would not see the killing. So that's just demonstrated that the killing was specific for the NY-ESO1 peptide. And we also measured the T cell activation. So as you can see that also the activation was specific to NY-ESO, to the presence of NY-ESO peptide. Now we tested this TCR-m-T construct in vivo. We can see here that in this particular study, when we transferred the TCR-m-T, okay, against NY-ESO1 into the mice that carries a tumor, and you can see that relative to the mock transduced T cells, our TCR-m-T construct, very potently inhibit the tumor growth, and actually eliminated the tumor completely after 20 plus days. So then we re-challenged this tumor with this model, these cured mice with the tumor again. Then in the previous treated mice, the tumor were rejected. They couldn't grow. So this indicates that the previous treatment by the TCR-m-T T cells has generated the memory. So the cured mice would actually automatically reject the replanted tumor. So with that, I'm going to summarize all the data I presented to you today. So, first is that we have been working on TCR-mimic antibodies in both the protein modality and also the TCR T cell modality of the Biocytogen. So this is because we wanted to leverage their unique capability to target intracellular tumor targets. And I also mentioned to you, described to you through the special cases that there's some unique features of our TCR-m programs. By being fully human antibodies, we generate those clones using humanized pHLA mice, knock-in mice, and we have a very effective immunization protocol and a rapid single B cell discovery and off-targeted testing processes. So the key feature for our TCR-mimic antibodies is that they are highly specific, and I hope that I showed the data that convince you that our clones were highly specific for the intended targets. And we have a number of programs. I described 3 of them, and we have other programs that are targeting other TAs, CTAs, or tumor driver mutations here. This is a slide that is a plug by the request of our BD colleagues. Just say we are very active. We are very open and active in seeking collaborations and licensing opportunities. And you can see that here's their contact, our BD licensing team, contact information. And if you are interested in our TCR-mimic antibodies, please feel free to contact them. And before I conclude, I just would like to thank, especially, thank our TCR-mimic antibody team that generated all the data that I described to you, and especially for the team leader, Dr. Jun Du, for his very generous support while I was preparing for this webinar. And also, thank you very much for your attention. Not sure if we still have time, but if we do, I would be happy to take some questions. Thank you.
Yeah, absolutely. Thank you, Frank, for that wonderful and very, very informative seminar. I know that we're running a little bit short on time, but I hope that you can stay with us for the Q&A session. And if not, no worries. We'll be able to share the recording with all the attendees. So let's get into it. So the first question was back on slide 18. I believe this was about the germline diversity of the MAGE-A4. So the question was whether interspecies homology conservation can influence the results. So, the very diversity between the different peptides.
Sorry, can you repeat it? This is slide 18, right? Jenna,
Yeah.
would you please repeat just the question part?
Yeah, so why does MAGE-A4 generate more diversity in terms of the heavy chain and light chain?
What is needed? Well, in our hand, really is you need to have a good antigen. Then, good mice strain, good antigen are key to generate the robust immune responses. And I think that is a key to generate the diversity of your clones. Clearly you are limited by the antigen you wanted to use, because if you want, for example, if you want to target WT1, it would have to be that specific peptide. It would have to be HLA-A2. So that part probably is similar across different programs in different companies, but it is what you used, okay, in the nuances in terms of the HLA and the peptide complex, and also the strain you use, that would give you the wanted diversity.
Okay, thank you. So on to the next question. So a specific question about the immunization with the HLA humanized mice. So do we use peptide only, or the HLA peptide complex when we perform the immunization?
Okay, we actually use a combination of those techniques. In other words, we did use the peptide alone, and we also used the peptide and HLA as a complex. And we also, depends on the targets, we also use protein immunizations and DNA and RNA immunizations. So we use a combination of those approaches depending on the targets.
Okay, makes sense. Thank you. So does the Ren TCR mice contain fully human TCR regions, so the human V and the human constant region, or does it only contain the V genes?
Yes, the fully human TCR mice
Mm-hmm.
contains the V alpha and the V beta.
Mm-hmm.
That's totally from human. I think the constant regions are from mice, just like our Ren mice. The variable regions are human, but the constant regions are from mouse.
Right. Okay, thank you. So given that the TCR-mimic platform gives high affinity binders, so what application would the Run TCR platform serve, given that these might be lower affinity?
Okay, we are very early the testing these mice. So we don't have a comprehensive view about the affinities yet. But since all, I mean, both of our Ren mice and the Ren TCR mice leverage the in vivo maturation process. We don't expect that our TCR mice would be much too low affinity. I say this just because I mean we don't have a lot of data to support that right now. But based on what we learned from the antibody side, when we introduced the fully human antibodies into the mouse, we got quite a similar affinities antibodies from those fully human transgenic mice. Uh, I hope I answered the question, but if I missed the mark, please let me know.
Sure thing. Yeah, we'll send follow-up emails to everyone just to make sure, there's no additional questions about the material or if you have any additional questions that arise after today's seminar. So no worries.
Okay.
So next question is, how do we control in the TCR-T model? How do you control random pairing between the endogenous TCR and TCR-M in the primary T cells?
Okay. One way is the our TCRm-T cells actually uses a mouse constant region, so that helps reduce the mispairing of our the our transduced molecules and the endogenous TCRs.
So I think this question is similar to the previous question. So I think I'll move on to the next question here. So what is the timeline for the immunization, sequence selection, in vitro and in vivo functional analysis if I provide a brand new antigen and HLA type?
Okay, that's also a very good question. Well, I would say typically it would be around 5 to 6 months from, let's say, you give us a target, you presented us with a target, and we do immunizations. We actually would do first evaluation of the target, then immunize and decide on the immunization strategy, and all the way to we would generate initial categorization data of those binders like specificities. That would take us about 5 to 6 months. If the targets is among the difficult ones, for example, they're very highly homologous to mouse target, then we would need more time. We would place a few tricks to make sure we can do the immunization in the effective way, and that would take some time. That might add say about 2 months to it. It depends really. I think if that's the case, let's have a conversation and we'll decide. We'll make sure we understand the need from you, that then we can propose a few different approaches. But in those difficult times, it would take longer. But barring that, usually it would take let's say around 5 months to 6 months time to generate the clones and do the initial characterizations.
Okay, so pretty fast. Thank you.
Yeah.
So a little bit of a open-ended question. So how about what are the parameters qualified as a good antigen? Well, you can.
Okay. A good antigen eventually is that you get a lot of clones, and here it's just a particularly very diverse collection of clones. These would be considered good antigens, but that's in retrospect, right? And so going into that, if the target is less homologous between human and mice, then you got a better chance. Okay. We this is because if they're very homologous, then their immunogenicity would be low. Okay. And other than that, it's just because this is a very, what I'm going to say, because you have to I'm trying to think of other ways to make a less immunogenic peptide more immunogenic, but our TCRm team may have some tricks. So if you usually the process, what we do is that you first let us know the target, and also if you already know the peptide from literature, then the TCRm team would do a thorough evaluation of the peptide, and they will come up with one or two strategies and talk to you. So far, as you can see, we have been able to generate dozens of clones for the programs I talked to you about. So these, you can say that they are good targets. They're very highly immunogenic, but we also believe that we have something going for us. We have some unique ways to generate those clones, to do the immunizations and generate those clones. So sometimes it's not only the antigen itself, but how you do it is also key.
Makes sense. Okay, so if using the mouse constant reagent to reduce mispairing, will you be concerned about the immunogenicity for the TCRm-T?
That could be a concern. These are, at least they are intracellular. So, but they, there could be immunogenicity issues. I'm trying to think whether, okay, so that I'm trying to think either Tsinghua team actually, they did the STAR T, okay? They actually performed a phase 1 clinical trial. At least based on that trial, there was no report about severe immunogenicity related issues. So that's n equals 1, but at least from their trial, there was no report of severe toxicity related to using mouse constant region.
Okay, thank you. So do we make our own peptide HLA complexes, or do we use an external company? And if so, who?
I'm trying to think. I know we certainly designed the way to make the complex, but I'm not sure whether we actually generated that in-house or we hired a CRO to express the specific forms for us. That we can get back to you after I consult with the TCR-mimic team on that specifics.
Okay, yeah, thank you. Stay tuned. So I think so there's a couple questions that I skipped over here. So So TCR-MT and TCR-T, right? They both recognize that the peptide HLA complex. So can you speak about any general advantage of one over the other, and are they off the shelf?
Okay. Well, the normal T cells use TCR, so
Mm-hmm.
that's actually people have been trying to do is to modify the T cell receptor, okay, to target the target they wanted to target better. But TCRm is very different. It's an antibody, so that's not the T cell receptor. So first, that's kind of a by that definition, these are two different entities. And then in terms of function, that TCRm, at the least for relative to the wild type TCRs, TCRm as an antibody, they tend to have higher affinities than the TCR than the TCRs binding to the peptide and HLA complexes. So that's why people need to engineer the TCR receptor to enhance the affinity. But by doing so, it might also create a non-specific binding situation. So you have to be very careful when you do the affinity enhancement. And people have successfully did that, but just you just need to have to be very careful. And another difference is that depends on the modality, of course, but in terms of solubility, TCR-mimic antibodies are a lot user-friendly than the TCR themselves. As many of you know, that T cell receptor are very difficult to manipulate in vitro, and they tend to aggregate. So it requires careful engineering to make them soluble. So that's these are two very different modalities. However, people are clever. They sometimes they make the hybrid is like the star key, or in our hands, the TCRm-T cell therapies. So people have ways to overcome the difficulties of TCR-based therapies, but in nature, these are two very different modalities.
Okay. Thank you, Frank, for that really, really nice discussion and presentation. I think that's all the time we have for today. But again, we're happy to follow up with any questions that we may have missed, or to clarify any of the material with you at a later time. So again, once again, thank you, Frank, for this great presentation, and have a great rest of the day, everyone.
Okay. Yeah. Thank you, everyone. Okay.
Thank
Bye
you.
now.